Hello everyone, in the previous video we discussed about the Krebs cycle and how we were able to generate an ATP molecule and actually two ATP molecules and 6 NADHs and 2 FADH. So just for a quick review, we studied in glycolysis that how a glucose molecule was broken down into two pyruvates, and along that process we were able to generate basically able to generate, if I'm not mistaken, 2 NADHs, and we were able to generate 2 ATPs. This was the product from glycolysis, and it occurred outside the mitochondria. It means it occurred in the cytoplasm. Now, after this process, we were able to generate these two molecules along with when the glucose, a 6-carbon molecule, when it was broken down into a 2-carbon molecule, we had pyruvates, and then they participated in the Krebs cycle where we were able to generate more ATPs and more NADHs. But before they were able to go into the Krebs cycle, they went into a series of processes, and each pyruvate went through a series of processes where it released carbon dioxide, and NAD plus was reduced to NADH, and along that process, when the carbon from this carbon, like you can pick up this carbon right here, it left as carbon dioxide, so a coenzyme A joined in and formed and called coenzyme A, and it joined with these two carbon molecules. Remember that this carbon left, so we had we basically were left with a 2-carbon molecule with a CoA enzyme called acetyl-CoA, an acid because of these two carbons right here. Now, since there are two pyruvates, so we released so the reaction ended up being you just multiplied by 2, so we had 2 NADHs and 2 acetyl-CoA, so we had another NADH right here. So just add another 2 NADHs. NADH. Now, then we discussed about the Krebs cycles. Now they are ready to go into the Krebs cycles. It enters into the Krebs cycles, it goes through a series of processes, and at the end, so we were able to, so just let me remove this, put it right here. Yes, so we were able to, from that process, we were able to generate 3 ATPs from each pyruvate, like from this pyruvate 3 and from this pyruvate 3. So at the end of the process, we were able to generate 6 NADHs, right? And along that process, we were able to generate 1 ATP molecule from 1 pyruvate. So if we're talking about 2 pyruvates, we generated 2 molecules of ATP. Now, in the Krebs cycle, we were able to generate another molecule which we called FADH2. FADH2. So this molecule right here, so we generated 1 for 1 pyruvate. So if you're talking about 2 pyruvates, so we basically generated, so let me divide this, so we generated 2 FADH2s. Now, so at the total end we had 10 ATPs, 4 ATPs, 10 NADHs. Just make a correction on that. Oh, it's gonna take me forever. Cut. Yep. That should do it. Now, where was I? Oh yes. We were able to generate 10 NADHs and 2 FADH2s and 4 ATP molecules out from the glycolysis and the Krebs cycle. Now, after this process, now we are at the third stage of cellular respiration, and all our main purpose is to break down glucose and generate ATP for our cells to perform functions of various kinds. Now, since we are in the oxidative phosphorylation, this is the third step of cellular respiration. Third step. First was glycolysis, second was the Krebs cycle. Now, so you might be thinking, okay, why is it called the oxidative phosphorylation, a really fancy name for a process? So at the end of the process, you will find out, and if you don't worry about it, I'll tell you. And so the oxidative phosphorylation is the third step of cellular respiration, and basically there are two processes involved, number 1 and number 2. So this oxidative phosphorylation is composed of two main kinds of processes, and the first process is called the electron transport chain. Now, from this word, from these three words, electron transport chain, so basically you're thinking, okay, electron is being transported in a chain, and it's somewhat similar. It's actually, the name tells you a little bit about what the process is happening. So as I mentioned, this process is occurring. So its occurrence is in the mitochondria. And in the mitochondria, we as we mentioned in the introduction to the cell part video, if you have seen them, so in that video we mentioned that we were discussing about that internal membranes, how the internal membranes of this mitochondria are called the cristae. My best attempt to draw mitochondria, and they are called cristae. So in the cristae, so if I were to just like zoom in a portion of this molecule right here, so if I'm drawing a membrane, and I'm drawing a membrane like this, and I have to color it in, so I'm just making it thick. Now, so this membrane right here, this is the single internal membrane, the cristae part of the membrane, so I have to color it in too, but that would take me a long time, so that's why I'm not doing it. Now, in the membrane, now this process is occurring within the membrane, and just to give you a little bit of idea of what's going on right here, is that the membrane is embedded, or it has proteins, complex protein structures stuck into it, and they have really fancy names. Like some of them are called cytoquinone, cytochrome B6F complex, and so if you're studying for higher biology or so, you might want to know these names, but just for the introduction of what electron transport chain is, so I'm trying to give you a little bit of overview of what this process is all about, and so I'm not going into all the details, and this is just a simplification of the process actually. So this is the internal membrane right here, and these are proteins attached into it, and there's one big protein. I'm just drawing a rough shape. Its shape is actually really strange. It's just a complex, like a protein complex, like big like this. It has no definite shape, but I'm drawing it in a flask-like shape to make it more obvious for the process which I'm about to tell you. So this is the membrane, and it has some proteins attached into it. Now what happens is the NADHs, which we were able to generate from the glycolysis and the Krebs cycle, and the FADH2s, these two molecules right here, when they reduce, when they oxidize, and remember, oxidation is losing an electron, or sometimes if you see hydrogen, it means the hydrogen along with that electron. So where was I? Oh yes. So when this nicotinamide adenine dinucleotide is reduced, it releases an electron. So if it was gaining an electron, it would be like NAD plus, and it had to gain 2 electrons along with 2 hydrogens, and we would eventually end up at NADH and plus a proton. It would eventually end up NADH and plus a proton. And so if this thing is being going reduced, so we just go in the opposite direction. So it's being it's being reduced, it's losing its electron. So these two electrons right here, when it loses this electron, these electrons are in higher energy state. And what do you mean by that? You might be thinking, by higher energy state, I simply mean that they are unstable. So they are like, you can imagine a person being angry all the time. If he's angry, so he's in a higher energy state, and you're trying to cool him down. So how do you cool down these electrons? So the key thing to cooling down an electron is that when these electrons the electrons leave the NAD pluses, they eventually go to these complex proteins. And what happens is, as they are going from this complex to this from this, so as they are going from one complex to another, they are eventually cooling down, or they are going from a higher energy state to a lower energy state. And eventually, when these protein complexes are carrying these electrons from one complex to another, from one to another, what happens is that these electrons, they gain the energy of the electron. So they are taking away the energy of the electron and pass it down to another, and then the other guy takes its energy, and until it becomes stable again. And so what do they do with that energy which they take? What are they doing is, actually, you have hydrogen protons here, and a lot of hydrogen protons over here. So if they are like 4, I will just draw 5 to show that there are a lot of hydrogens. 1, 2, 3, 4, 5, and let me draw one more. And let me get another color. So there are a lot of hydrogen protons here and protons over here. So what happens is, when they gain that energy, these complexes are able to transport hydrogen protons from the inner membrane, from the membrane here, and I mean the internal area of the inner membrane, to the outward area. So they are transporting it actively. So what do you mean by actively? And what it means, like, there are sometimes called the passive transport, and sometimes they're called the active transport. Such as, like, when you're talking about active transport, but basically it means that you need energy to transfer it. Since we are going from a place where it has low amount of something to a place where it has a higher amount, you can give this example like this. But if you get a perfume bottle and open it in the corner of the room, so the corner of the room where the perfume bottle is, it has more perfume in it, and eventually spreads up to the entire region of the room where the perfume is less. So now we're doing the exact opposite. We're going from a place where it has low amount of something to a place where it has higher amount. So you need energy in order to do that. You're going against something, so you need force or power or energy in order to perform that function. And what are these complexes are doing that thing right here. The complexes are able to put protons from the place of less concentration to a place of higher concentration. And if you love physics, or if you love some kind of, like, if you love physics and you're into mechanics and all that, you will eventually get this idea. Oh, if it goes from a less to a higher, so it must gain some energy. And what kind of energy would that be? If you're changing your position, you gain what kind of energy? And you gain the energy you gain is called potential energy. And this is what's happening right here. As these protons are moving from a less to a higher concentration, you can imagine there are a lot of protons on this side of the membrane. So they have a lot of potential energy. So as it's going from the electrons are going from here to here, from here to here, it keeps reducing its energy and giving these energy to the complexes, which are able to generate protons up into the membrane. So you might be thinking, okay, what's the main reason of that? And so actually this is the reason why we are able to generate more and more ATP. Now, as it goes and as it stabilizes, the last of the electron acceptor would be oxygen. Now, oxygen, when it accepts these 2 electrons from the NADH, remember I showed you right here there are 2 electrons. So if an oxygen accepts 2 electrons, so you might be thinking, what kind of notation is that? It's basically half of molecular oxygen, which means an oxygen atom. Now when it gains what was the thing? Oh yes, when it gains the electrons, and it has some hydrogen protons in the internal membrane too, so it gains 2 electrons along with 2 hydrogens. So if you gain 2 electrons with 2 hydrogens, you'll eventually end up with water. uh, Water, right? H2NO. So you eventually end up with water. So that's what we learned in the first video of cellular respiration, that cellular respiration is the breaking down of glucose into carbon dioxide, water, and ATP. So we eventually learned where we get the carbon dioxide, and now we are here. So we find out where water comes from. Now, okay, so you might be thinking, okay, what was the main reason why these hydrogen protons are up there? And the main reason is that, so now they cannot come back, so they have a lot of potential energy, you can imagine. So these potential energy are somehow want to be normal again, to go into their normal state, so they are not comfortable in that position. So what they do is, so now a membrane doesn't allow them to go into the back into the membrane, because basically the complexes don't have enough energy to do that. And so what they do is they go through this complex right here, which we call the ATP synthase. ATP synthase. And you can imagine by its name, it's pretty obvious that it makes ATP. And what happens is that when these high potential energy stored protons go into the ATP synthase, it eventually, literally, sorry for my pronunciation, it literally causes this protein complex to rotate. And as it rotates, it has ADPs. So let me draw it. It has ADPs plus phosphate groups already present in it. So what happens is they spin it. You can imagine as like when you're making a milkshake, like you're putting all kinds of stuff in there, and then you press the button, it mixes everything up. So it mixes the ADP with the phosphate group. And when you add a phosphate group to a molecule, that's what we call, ta-da, phosphorylation. So when you add a phosphate group to a molecule, you call it phosphorylation. Now, you might be thinking, okay, where does this oxidative come from? Actually, it's from redox reaction. Redox reaction literally means like it reduce and oxidize. So, okay, where does it reduce? It reduces when you lose the NADHs. So you're losing the electrons, like you're going backwards. So you're doing, when this process happens, you're basically reduced. So that's where the re comes in the redox part. And when you gain electron by different electron complexes, and they eventually lose their electrons, so it's called the reduction and oxidation reaction at the same time. They're happening at the same time, so you call them redox. So the oxidative part comes from this, the redox reaction. And this process of these high energy protons and going into the ATP synthesis and making ADP, this process is called osmosis. And this part where electrons go from one complex protein, and complex means it has really, by the word complex, it means really complex, really difficult. It has all the proteins in it, and it's sort of weird looking shape. And this part right here, where it goes from one electron to another electron, is called the electron transport chain. The electrons are being transported. Electron transport chain. Okay, and what else? Oh yes, another thing which I wanted to mention, I actually saw it in another video, and hope you guys will find that useful too, is that each NADH molecule is responsible for generating 3 ADP molecules. So if one ADH molecule is reduced, the electrons of that energy are able to have enough energy in them to produce 3 ADP molecules, and each FADH2s are able to generate 2 molecules of ATP. So So I think in the previous video I mentioned that we end up with 38 molecules of ADP. So if you just make it and do the maths and calculate it, so we have 10 NADHs. For each NADH we have 3. For so 10 we have about 30. And for we have 2 FADHs here, right here. And so if we are talking about each FAD is 2, so 2 makes it 4. So plus 4. And we already had 4 ADPs, which we were able to generate 2 from the glycolysis, 2 from the Krebs cycle. So we eventually end up with 4 ADPs, and the total would be 38 ADPs. And here you go, we have our 38 ADPs. So this was pretty much it. So hope you find that useful, and in the next video we're going to talk. I'm planning to make a presentation on the photosynthesis, which is a really amazing process. So hope you found that useful, and so see you in the next video. Bye bye.